I'd like to introduce Dr. Beate Ritz. She is an expert in the field of epidemiology and environmental health sciences from UCLA. We heard her talk at the World Parkinson down in Portland, and we're very, very interested in bringing that information back here to all of you in British Columbia. She is going to be discussing the relationship between genetics, the environment, and Parkinson's disease. So please join me in welcoming Dr. Beate Ritz.
Well, thank you for inviting me. I'm really thrilled to be here. I actually just realized it's 20 years since I started to do Parkinson's research, and it seems like yesterday. So I'm an MD Ph.D. I decided that I wanted to study disease not by seeing one patient at a time, but by studying diseases as they come upon populations, and that's actually the word, it's epidemics or epidemiology. And you might not have thought of Parkinson's as an epidemic, but in a way it is. It's the second most common neurodegenerative disease of elderly, of people over 60. Of course, we have the young onset Parkinson's patients as well, but really, you know, that's when we start worrying, is when we get older. So why did I do this? Well, I actually had a personal story where I started as a young psychiatrist in the University Hospital of Hamburg. I'm German. And my boss developed Parkinson's disease. And he was one of the most inspiring people I knew. And within the 2 years that I worked with and under him, he progressed severely, and he gained almost 20 years in age in front of me. And that was in the early 80s, when I asked, well, what is it that causes Parkinson's? Nobody had an answer. And when I looked around at research, there was none. And honestly, there's still very little population research on Parkinson's disease out there. And I believe one reason is that we don't even often know how to find the patients. Cancer has been, the war on cancer was declared in 1970. At the same time, in the US and in Europe, we started having something called cancer registration. And even though that might sound really bad, it was actually the best thing that could have happened, because it made people like me, researchers, be able to contact patients and to find them and to enroll them in studies and to do all this wonderful work that the cancer community did and is now really using in treatment, in prevention. Everything we know about cancer was partially due to that effort. So I have received NIH funding, Michael J. Fox, Department of Defense, and private donor funding over the last 20 years. But I'm standing here after the 20 years for the first time in my life not having $1 of Parkinson's funding in my research lab. And that is because the institute at NIH that had supported me for 20 years, which is the Environmental Institute, yeah, you know what I'm saying, right? Unfortunately, however, it wasn't just the political climate, it was actually something else that happened. The leader of this institute, when I was a junior researcher at UCLA in the mid-90s, the then leader of the institute saw Parkinson's as the new frontier because he saw exactly what I saw. There was very little population research out there, and he wanted to build this up. So Kenneth Olden, the then director, funded Parkinson centers that this institute was funding over many years. And they got defunded, and they got defunded because other pressing needs were bigger. And we have children's centers within this institute now, many, but we don't have the neurodegeneration ones anymore. So, you know, tides come and go. Sometimes it's the children, sometimes the elderly. But what it taught me is we really have to keep our voice being heard by government, by agencies that fund this kind of research. And even so the tides come and go, you have to keep pushing, because what once existed may not exist tomorrow anymore, because, you know, somebody else's voice was a little louder than yours. So I just want to encourage you, do your best to make sure that, this doesn't go under in terms of interest. So I have this slide that I really don't need to show you because you all know what Parkinson's disease is, but it does remind me to emphasize that really only in the last 5 to 10 years, we went from defining Parkinson's disease as a motor disease, right? A motor dysfunction. And the symptoms, tremor, rigidity, akinesia, postural, reflex impairment, we call them TRAP, right? And you probably all feel sometimes just like that. But what we really learned is that before all of these motor symptoms emerge, we have a lot of non-motor symptoms to deal with. And they're often actually happening prior to the motor symptoms emerging. Some of you may have had depression many years before Parkinson's happened. Some of you may have had sleep disorders, loss of smell, peripheral autonomous nervous system disorders, constipation. We now know from population research in large populations where we followed people, like 100,000 people over 30 years. That's what epidemiologists do. They follow cohorts of people. We learned that constipation may be 20, 30 years happening prior to your motor symptoms. We need more of this kind of research. We also learned what happened in the brain, and we learned that from our basic science research colleagues, and that's what you see here, these little blobs there in the cell. They're called the Lewy body. And the Lewy body was something that pathologists had found 100 years ago when they stained the brains of Parkinson's patients, right? But we did not know until in the mid-90s, when I started my research, what was actually in those Lewy bodies. And one of the proteins was synuclein, and I'll come back to that protein. And then, of course, population-based studies were rare, and pretty much all we knew when I started out was that there was an age-dependent incidence increase in Parkinson's, and that's the little figure you see below there. That's actually age starting from 50 to 90, and you can see that it's almost a logarithmic increase in the age relatedness of this disease. And so we know that aging definitely has something to do with this disease. However, we are usually asking aging, we're all aging, right? But some develop Parkinson's, others don't. So it can't just be regular aging, even so age is that main risk factors. So what we really want is to find causes and treatments. And my medical colleagues who are still seeing patients have an advantage. They can do trials. Me as a population epidemiologist, I have to watch people. I have to watch them over time, for long periods of time. And while I watch them, I have to be very careful that I don't fall into traps, into biases, that I do not connect things that really have nothing to do with each other, except that they're happening simultaneously. It's the story of the stork that brings the babies, right? We know that where there are houses and storks can nest, they're often young couples and they have babies, but it's not the stork that brings the baby, right? So it would be nice in some nice story. So we want to stay away from identifying associations by proximity in time and space, and we really want to know what is happening causally, right? And that's very difficult, but that is one thing I teach my students. I'm a teacher of methods at the School of Public Health in epidemiology, teaching them how to do these observational studies so that they don't make all the mistakes that are so easy to make, and that they really can identify causes. And hopefully, in the end, we can also identify treatments for these causes. So my geneticist colleagues were much more successful in terms of, in 1997, right when I tried to get my first Parkinson's funding and finally got it, actually it was a private donor who gave me a $35,000 check on Christmas. It came to my department, and it paid for several students who worked with me for more than a year, and we finally got big NIH funding after that. So that was a really good investment. But in that same year, a geneticist found a gene. And that's the story coming back to synuclein. It was a gene we didn't know at the time that was actually a synuclein gene. And synuclein, remember, was the protein that clumps together to form these Lewy bodies in your brain. And that was 1997. And where they found it was actually in a large family that was located in southern Italy. And then they could also trace back roots to Greece from immigrants who had immigrated to Italy. And through this family tree analysis and the then new tools in genetics, they could actually identify where that gene was. And they really, and finally, so I was just starting out as an environmental population person, and there somebody found the gene for Parkinson's. I should have closed shop, right? We knew what it was. Well, it felt that way, but I said, maybe it's not the whole story. And guess what? It wasn't. That was the only family worldwide that ever had that mutation, okay? There are other mutations in other families, and there are duplications and triplications of that protein, but never again have we found another family like that with exactly that specific mutation. Well, it taught us one thing. It taught us that there are genes and there are mutations that can cause what looks like Parkinson's and what is Parkinson's, but there must be a lot of other things out there that are also important, but they also involve the same protein, because all of you with Parkinson's have these clumps of that protein in your brain. Okay, so clinicians, of course, want to know what prognosis their patients have and what they could do to improve the prognosis and how they could treat the symptoms. So that's another set of interests, but I think the geneticists and myself as the environmental epidemiologist, we can help them do this. And then I want to study general population Parkinson's patients, because I believe that if you only are studying people who come to a specific special clinic, you don't get the whole picture. You know that every Parkinson's patient has something in common with the next patient next to him, but they're also different in many ways, right? And they are different not only in types of how their disease looks like, they're also different in what might have caused them. And I want to tease out what is similar and what is different. And by doing that, we have many different stories, but in the end, we can probably help all of you much better. So this is the slide I put together this morning because I thought I have to tell you something about those genes because everybody will be talking about them. And I already said synuclein, that's SNCA, that's the short version of it. And there are many others. Parkin, that was the second mutation found. And since the first one, we already had a word for that gene, synuclein, or that protein, only the second genetic hit, and that was very shortly after the first one, was called Parkin. And then geneticists said, now we really know. But again, it turned out it was a very rare occurrence in Parkin too. But finally, in the more recent years, we found that actually Gaucher disease, GBA mutations, put you at risk of Parkinson's. And those are much more common in certain populations, in certain Middle Eastern populations, right? But they are a risk factor. Not everybody who has these mutations, actually few people among them who have these mutations will ever get Parkinson's. So the big question is, what prevents those who have the mutations from getting it? That's my question, right? Shouldn't we study them, the ones who have the mutation and then don't develop disease, why they don't? And I think some of my colleagues are starting to do that. The same with the LRRK2 mutation. It's also a North African, Middle Eastern mutation that is very common in certain populations. In the US population, it's less than 1% that have these. So what is common? These are all very rare mutations. We now think that less than 5% of people with Parkinson's have any of them, okay? So geneticists are happy to have found them because they tell you something about the disease. They point you to a pathway. They point you to a protein. We hope that we can intervene and maybe declump that alpha-synuclein eventually. But honestly, until today, we don't even really know what synuclein does. We know it's in the brain, we know it's in the synapse, we know that it's in other parts of our body, but we have no idea really what it does. And we have no idea how we can actually make it not accumulate in the way that it forms a Lewy body. So have we gotten other tools? Well, what have we learned about Parkinson's from looking at populations? I already told you there's a family history in some patients. That's the 5% of family history-derived Parkinson's. Being a male is not a good thing. In the sense that you have a higher risk of developing Parkinson's. Aging is not a good thing. We all want to age. We all want to get old. I can't tell you not to. So again, there's not much we can do. But then do we know something else? And that's where I started to look at pesticides. And that is a very specific story I'll tell you in a second. Some other colleagues have been studying solvents and metals. We also know that there is a head trauma risk. Muhammad Ali is known. He had lots of head trauma. However, it's not really clear yet whether that is really a big risk factor at all. And then we have on the other side, see, I say causation or reporting bias. We are always, I told you, we have to avoid these biases. Are patients just telling us more about these things, like head traumas they had because they want to find a cause for their disease, right? Or on the other side, you see protection. So you may have heard that people say, well, caffeine is protective, smoking is protective, anti-inflammatory drug use is protective because it reins in inflammation in the brain, physical activity, diet, right? These are all no-brainers, physical activity and diet and NSAIDs. But why is smoking protective? Well, I still don't believe it is. I actually am with the old neurologist who told me, so, Dr. Ritz, do you know when Parkinson's starts? And I try to find that explanation, right? And he says, I tell you, I know exactly when they stop smoking. Okay? And so I kind of thought in my population studies, well, what can I do to really find out that that's true? And there is no easy answer, and there's probably no way to really find it out. But I did ask patients in my study how hard it was to stop smoking. So, and at what age they stopped, and whether they needed help stopping smoking. And I saw that those who had a very easy time smoking were at higher risk and got their Parkinson's first. Okay, compared to others who tried to stop smoking, who never developed Parkinson's, my control group, those who had it easy were on their way to Parkinson's. And I interpreted that as not responding to the nicotinic rush anymore. So one thing that your brain does is, change all sorts of circuitry, all sorts of things that happen in the brain. And one is that it doesn't respond so well to nicotine anymore. In epidemiologic terms, we call that reverse causation, right? So it's not the smoking that stops Parkinson's, it's the Parkinson's that stops smoking. You will still find colleagues of mine who say, Beate, that is much too pessimistic. We need to show that there is some substance in that 3,500 chemicals in smoke that is actually protective. And I wish them good luck. And, I don't want to be negative. Maybe there is that one substance in there. But caffeine, I'm a little more hopeful, and I'll show you why. And, of course, a good diet and physical activity can only help. And I show you actually data that we see that it does. So pesticides was really in the mid-90s when I started, pesticides were my field of interest because I live in California, I live in LA, yes, the air is bad, but I also live south of the Central Valley, and the Central Valley of California is among the biggest agricultural producers in the US. In terms of a very varied agriculture. So we have fruits and vegetables and cotton and alfalfa and rice, and I can go on and on. And how are we farming this? Well, we're farming this in 4 seasons because we have the sunshine. And we are farming this by using about 640 different active ingredient pesticides, about a quarter of all pesticides applied in the US. And that's a lot. And we've been doing that for 40 years. California is a little in the forefront of environmental things, legislations. So in the early 70s, they actually produced a system called the Pesticide Use Reporting System, so that farmers had to actually tell the state what they're applying, when they're applying it, and what they're applying it on. And I was honestly the first person to ever use this systematically in population research. And that was the $35,000 check that helped me do that. I retrieved that data and I had my students work on it. So why are pesticides important? I tell you, yes, for the farmers in the Central Valley, they are important. Right? But you who live in Vancouver or somewhere nice where, maybe nobody sprays pesticides on your golf course, heavy herbicide use on golf courses. Don't kid yourself. But the statistic from N. Haynes, that's the big survey done every 5 years in the US of a random sample of representative Americans, found that almost 100% had a certain pesticide called chlorpyrifos in their urine, meaning they had breathed it in, eaten it, and excreted it. Okay? We can test you. You will all have a metabolite of DDT in your blood, all of you. Because we used this widely in the 60s and 70s, and it sticks with you. It's in your fatty tissue. We know that this herbicide that replaced paraquat, which I will talk about in a second, glyphosate, that has made the news a lot because of carcinogenicity. We know it is found now in 100% of soy products, in 20% of all breads. It's still believed to not be so toxic or toxic at all, but IARC declared it a carcinogen. So we cannot escape these agents. Whether we really ingest them, breathe them at a level that will harm us, like the farmers, right, who have heavy use and heavy exposure, hopefully not. Let's hope not. But some among us may be specifically sensitive, others, wouldn't. So, what was it that got me interested in pesticides? It was this story, and it's a California story. This California story goes that in the mid-'80s, all of a sudden, in south of San Francisco, in the Santa Clara region, where Google is and Apple, just so you know, there was a different cottage industry at the time that produced synthetic heroin in the garages and not computers. And that was because of the legislation in California that only made a certain substance illegal, but not designer heroin. So you could actually produce it, and because it hadn't the chemical structure, you could get away with it, that it wasn't illegal. And so these smart young people produced these designer heroines, and then all of a sudden there was an outbreak of about 30 young people showing up, under 30, showing up with Parkinson's, instantaneous Parkinson's. And you all probably heard about the story. It's the frozen addicts. And Bill Langston made that very well known, right? And he was the one who found them and actually diagnosed them with Parkinson's. And that was a real breakthrough because they also found what it was. It was a contaminant. It wasn't the heroin or whatever they were trying to make. It was a contaminant that was called MPTP, a methylpyridine. And this contaminant of the designer drug was able to go through the blood-brain barrier, be converted to a very toxic agent, MPP+, and you see this chemical structure. And that was what killed the dopamine neurons and did it instantaneously. The good thing is we now have this agent to actually cause Parkinsonism in animals, mostly in primates, and to try treatment on these animals. Poor animals, but that's our chance to try out treatments. But look at how it looks like. It looks very close to another chemical below that's called paraquat. That's an herbicide, meaning it's a weed killer, right? A weed killer is not supposed to do something neurotoxic in your brain. It kills weeds. However, look how similar it looks to MPP+. And actually, MPP+ was just about to be introduced as another weed killer when they found this. Luckily, they didn't. It was supposed to be called cyberquat, okay? So when they found that paraquat looks so similar, colleagues who did animal studies tested them out in the lab and were able to create what we now know as the paraquat mouse model of Parkinson's. So we have these animal models of our disease, our favorite disease, and one is the model that causes, in monkeys, the Parkinsonism. That's the MPTP model from the heroin derivative contaminant. And then we have the paraquat model in rodents, and we also have now one based on rotenone. That's another pesticide. It comes from an African tree root, and that it's natural. The tree defends itself against insects, right? And that natural pesticide is sold as an organic pesticide. We, however, can use it in mice and rats to cause Parkinsonism. That is very similar to what you see in patients. So when I knew all of this, and I knew that California had actually done this big effort of putting pesticide records together, I went to NIH showing them I can get these records and I can do some work with them, and please give me money. And after 3 attempts, they finally did. And I was funded for the study that now has collected data on more than 800 patients in 16 years. We started in 2000. And in about 1,000 population controls. And my movement disorder colleagues go out there, drive 4 hours, see the patients, assess them. We bring back blood, we bring back urine. And we then have the patients on the phone with us for hours telling us what we want to know about their lives. And so this is, by the way, the use of paraquat that I just mentioned in California. And where you see all the really dark spots, that's heavy use since this registry happened, that's where I'm doing my study, okay? In that you see the Central Valley by the use of paraquat. And this is how we selected controls. So we mapped people's homes, and then we randomly selected. These are numbers because we selected parcels. These are parcel numbers. And then we selected these homes, and we went to the homes, and we asked people to enroll, the people who didn't have Parkinson's, to enroll. The Parkinson's patients we got from clinics and from hospitals, and we went around churches and had news announcements because nobody had a registry and could tell us where to find the patients. It was a huge effort. And then the controls, we went home to home. We did 15,000 home visits, right, for these 1,000 enrollees. 15,000 because we had to come back. We sometimes came 5 times. And then, unfortunately, or fortunately, 80% of the people who lived in these homes were not in the right age range. We didn't want young families, we wanted elderly, right? So, huge effort. You can see why you really need NIH to get involved with big funding. This was big funding. This was $5 million, okay? So what we did then, my students had started, was to computerize the pesticide use. And you can see here the squares were what the pesticide use report system was telling us about what pesticide was applied in what square. We then also digitized, computerized maps. And then we overlaid the maps of the crops with what the system about pesticide use told us, and we could actually define much better where the use happened. So we don't have these squares anymore, we have fields now where the stuff was applied. And we did that by using these paper maps on the left and turning them into these maps on the right that are computerized. And now we know where the almonds are, where the peaches are, where the field crop is, right? And when we then have the addresses, and this was really tough on our patients, they all had to remember addresses. when we knew where their address was, we could then draw these circles around their homes and see how close they had been to pesticide applications. And we did that for all of the patients and for all of the controls, for the people who never developed Parkinson's, because in our type of research, you need to compare patients with those who did not become patients in terms of what they were exposed to, or else you don't know. Maybe everybody is pesticide exposed, but it doesn't cause Parkinson's. So we need that control. And this is how in California, in the Central Valley, up in the left upper, you see a little yellow plane. Maybe you don't see it. But the white stuff you see, that is spraying of pesticides, okay? And you can also see that there are homes underneath because there's some drift. So this is certainly an application method that causes lots of exposures. And this was a study EPA did in the Central Valley in the early 2000s, and all I want to tell you is that they had monitors on 3 elementary school rooftops and measured pesticides, and on the right you can see the measurements in the bars, in the blue bars, and what you estimate from the pesticide use report system by drawing these circles around these communities, what was applied. And you can see what was applied turned up on the rooftops in the measurements. So you can actually measure this stuff. So we have our animals, and in animals we know paraquat together with a fungicide, MANEP, causes Parkinson's-looking features. And my study was the first one to show that when people were exposed, using this exposure assessment system that I just explained, when they were exposed to both paraquat and MANEP at the higher rates, their risk of Parkinson's increased 70% to 100%, almost doubled. Again, we are in the Central Valley. We are where all this stuff is being applied. So I told you I actually like genes as well, but I like them for a different reason. I like them because genes sort us. And it's not the genes that give us Parkinson's, it's the genes that help us detoxify. That's why not every smoker gets lung cancer, because some people are lucky enough that they have a biology that gets rid of toxins faster than others. And this is what I used in my studies. I used these dopamine transporters to sort people into different groups of people who would transport paraquat into the cell faster or slower. And this is what we saw. When we sorted people according to these risk alleles, on the left you see a study from Washington. So this is a colleague who did this in apple farmers. And you can see that bar in the back, the exposed. These people are exposed and have these dopamine transporter risk variant genes. The ones who have only one, their risk is only slightly increased, and the ones who don't have any of these genes, you can expose them, they don't get Parkinson's, okay? And this is actually one of the very first replications that we were able to do in any epidemiologic study of pesticides. On the right, you see my results in 2009. So my colleague came out in 2006 with his results, and I finally got around doing it in 2009 because we were still collecting patient data. And you can see I completely reproduce what he showed. It is not the toxin alone, it is not the gene. The gene actually doesn't give you Parkinson's at all, but the two together increase your risk, so that in the end you see the 4.8 up there and the 5, a 5-fold increased risk, okay? That's not minimal. This is another example of a transporter of certain pesticides across the blood-brain barrier. Left is a French study, right is my study. We see the same thing. And this is another example. And this example is important because remember I said chlorpyrifos was found 100% in the urine of the US population. Chlorpyrifos is that one in the middle with the black and then the big gray in the back. And so we have zero exposure and low-high exposure of chlorpyrifos. And then what gives us the depths is the metabolizing ability of an enzyme that turns this organophosphate pesticide into a less toxic agent. And you can see that you have the increased risk, that steep increase among the slow metabolizers, the people who cannot turn that toxic agent over fast enough. The others have an almost non-existent risk. So who among us has the slow metabolizer versus the fast? We know that this enzyme has a 40-fold difference in populations in activity. 40-fold, that's natural in our population. So you might be 40 times faster than I am in metabolizing. This is another example where, so the slide before showed the exposure assessment I did with the tools I had was the pesticide use report system and the agricultural use. This, we asked people to tell us what they were spraying in their gardens and homes. And then a student for a whole year tried to identify exactly what was in those products by going through products lists and names and types of insects, etc. And you can see that that bar that says 2.6, that is a 2.5-fold risk increase, were the agents she identified that were organophosphates. And in those people who again had the slow enzyme that couldn't turn over these organophosphates fast. And these organophosphates, a lot of them were chlorpyrifos because we actually used that indoors for pest control in the 90s. So we did a lot of these studies, looking at different pathways, looking at different pesticides, and I wrote one big overview article a year ago that you can find if you want, and this is one picture from it showing all the different pathways. We also looked at brain injury, and we found that, yes, brain injury gave you a slightly increased risk in about a 50% risk increase in Parkinson's. But when you were a farmer and also exposed to paraquat, that's when your risk was really, really high. It was threefold. So we have now two risk factors coming together. You have your brain injury or your head trauma, but you also then are paraquat exposed. Again, you know, it's not one thing that causes disease. It accumulates over a lifetime. And what we have been doing lately, and this is the last 2 minutes of my talk, I hope, is we followed these, or we are trying to follow these 800 patients over their disease. So I just told you lots of things that can actually cause Parkinson's, maybe prevent Parkinson's, but we also want to know what can slow down progression. And this was the toughest thing I've ever tried to do, that is get money for these progression studies. And I've been trying for 5 years. I still don't have money. One center gave me money in 2007, and we followed 250 patients with that money. And since 2012, I've been trying to follow the other patients, and there is no money. However, Dr. Bronstein donates his time, and I have lots of volunteer students, so he keeps going back to see these patients, but we can only do minimal. However, the first time around, we had money to actually do this, and we were able to follow 250 of our 370 earliest patients. And this is what we were able, and 64 were already dead before we could follow them, because we had to wait 7 years before I got money to follow them. This is one thing we found. We found that synuclein, we're getting back to that synuclein protein, right? We have common variants in this protein in something called the promoter region. And when you split patients up into those with certain variants in that region of the gene, you can see that their motor decrease is faster when they have more of these mutations. So this is one of the first papers showing that there's actually possibly a genetic risk with having more of these mutations so that your decline is a little faster than in others. We also found that the common ApoE4, you all heard about ApoE4 because that's the main risk factor for Alzheimer's. It also works in Parkinson's. The cognitive decline among those who have that ApoE4 variant is faster in Parkinson's patients than in those who don't have it. Tau doesn't work in the same manner, and we all had our money on tau. What we also were able to do recently is use everything our genetic colleagues have actually found as so-called SNPs, single nucleotide polymorphisms, which are variants in your genes. And they scanned the whole genome, and they found these variants, and they linked them to Parkinson's, and there are now, I think, 27, some of them. And you can see that plot. It indicates with these weird numbers and letters all these different mutations. And when you line them all up and you generate a score of how many of them you have, you can actually also predict, and this is not even out in the literature yet, we are sending this paper out in the next few days, we can actually predict motor decline and also cognitive decline with these. It's a risk. It's not deterministic, but we can show that you're more likely to decline faster. Why is this important? Nobody wants to know this, right? Except the people who do treatment trials. They want to pull out the ones who are at higher risk of declining fast because they can see then much faster whether their medications work. So this is important for that reason. We need to sort patients into piles of, okay, these people are the highest risk of having fast motor decline, of having cognitive decline. Now let's try the medications on them, okay? We also found that my favorites, the organophosphate pesticides, influence cognitive decline. Those with the highest exposure had the fastest decline. We found that some vitamin D genes seem to protect you from cognitive decline. Others might be not so good for you. And we found that coffee is actually a good thing for progression. So all go and have your coffee. Those who never drank coffee progressed faster in terms of cognitive decline, and they also died earlier among the patients. So this is just comparing patients to patients. Tea helps as well. What else are we? And these are all data we haven't even pushed out into the literature yet. And the only reason why we can do currently analyses is because my students got fellowships because the money ran out to do these analyses. We're sitting on a pile of data. So we're finding that education is good for you. Your cognitive decline is slower when you have been educated. It also means keep your mind active, okay? Anything you can do to keep your mind active will be good. Sleep is important. We know that too little sleep is no good, but actually what we are finding is that if you have been sleeping more than 10 hours or you report sleeping more than 10 hours, it's actually a bad sign for your cognition. And we find that wine drinking, as usual, protects against overall mortality. So, but have that one glass, right? The one glass is probably what you want. And physical activity. We all did our exercise a minute ago. We actually have been showing that, you know, whatever you can do early in life, late in life, it all works. And that's one of my students. My time is over. One of my students who's coming for this conference will show her posters on exercise, and this is one of the things she shows. If you had low exercise your whole life, you never changed, your risk is the highest. But if you went from low to high, you're reducing your risk by about 40%, and if you went from high to low, it's still better, and if you're high, you reduce your risk the best. All right, I'm skipping this. So what we need is a strong scientific story that is stimulating, that will get us intervention and treatment trials, or as public health officials, will get those toxins out of the environment, right? And that's my job. And I'm not talking about, so the registry was one hope that we have that will help us do this. And these are really the people who did most of the legwork. And you see Dr. Bronstein and Bordelon who drive for hours to see the patients in the Central Valley, and lots of generations of students. These are old pictures, some of them. Thank you.
Fabulous, frightening, and fascinating. Thank you very much. We do have, 15 minutes for questions. We have both Caroline and Miriam with roving mics. Run one right up here. I'll try to go from side to side.
Organic tofu, because I don't eat red meat,
Yeah.
and I eat some fish and maybe a bit of turkey today. I eat lots of organic tofu. You mentioned that 100% of soy products may be suspect.
Right. I mean, if it's grown organic, it shouldn't have any pesticides on it, right? Because we hope that's correct. Actually, the only real story I know about this is a colleague of mine doing pesticide research at Harvard. He asked women who had 2-year-olds to feed them for 2 months organic food only, and he tested metabolites of pesticides in their urine before and after, and he found a lot of them before and none after. So if you can afford organic food, I know it's not the cheapest, it might work.
It's just a quick, simple question. A copy of your presentation available that we could download off the website or something similar?
I will have to take out some slides of things that weren't published yet, but the rest, yes,
Thank you.
definitely.
Great,
Yeah.
thanks.
Just curious, before the use of pesticides, you said they've been using it for 40 years. What are the, what percentage of people had Parkinson's then, and what compared to now after
Right.
40 years of pesticide use? And then the organic pesticide that you mentioned, is that I didn't quite catch the name of that one.
Rotenone. Yeah. You're right. Parkinson's is older than pesticide use, although our farmers were always inventive, right? Before there were the chemical pesticides, there were arsenicals. Arsenicals might not be good for the brain either, we know that. And they were used in the 19th century already. And there were the so-called organic pesticide, chrysanthemum, pyrethroids. Nicotine was a pesticide, right? Now we have neonicotinoids. So farmers always kind of learn from nature what is toxic, because that's what plants do. They defend themselves from being eaten by generating pesticides themselves. Of course, the modern farming is using them in high concentrations and get us in contact with them in high concentrations. But I'm not saying that pesticides are the only cause of Parkinson's. Don't get me wrong. There are many causes, but it may be one of those contributors that, the last drop that brings the barrel to flow over. And we don't know really what the change in Parkinson's incidence is, because nobody has tracked it. Different from cancer, where we have the numbers every year, we don't really have that for Parkinson's in the US. I think British Columbia might be better off because you have electronic health records and you could actually look over time what's happening. Yeah.
That kind of leads into my question. I was wondering if you use at all the artificial intelligence to streamline this kind of research.
I would love to do that. Yes, so we know that the cancer registry is already using these word crawlers for electronic medical records to find the words in medical records that point to cancer. But of course they got it easier because pathologists are seeing every cancer and they are reporting to the system. But for Parkinson's, that might be one of the ways to go. So we need, funding to do this big data push, where we can crawl through records. And also, in Denmark, I was able to do that. I didn't show you those slides. But I have a big Danish study with about 2,000 patients. And they have medical records you are allowed to look into and then search for. And we can actually gain a lot of insight from the medical records. We can also link the records to a pharmacy database that allowed me to look at what people took before they became Parkinsonian compared to the people who didn't. And for example, we found that a calcium channel blocker might be protective. It's one you use as an antihypertensive since the 1960s. It's something we cannot do in the US because we cannot link these databases. The Danes are very trusting of their government, of their researchers. They let you link these records. That's where I could look. But this is so far the worldwide only study in humans showing that possibly these calcium channel blockers might protect neurons. As colleagues of mine have shown in mice is the case. But mice and humans don't always, translate very well. There's now one other record system where we can look again, and that's in Taiwan. And I have a Taiwanese postdoc who is trying to get money to do that, to look in a second human population to see whether it holds up. I heard about it, but I'm not completely sure that I know what it is.
They could probably find something for Parkinson's. This study. I don't know, this is just recent.
Yeah, no, I wouldn't be able to comment on that. Yeah, but I can tell you we epidemiologists have always been big data people. We are data hounds, right? But really. But what we also learned is that just throwing a machine into on a data set or having no hypothesis, having no structure to what you're looking for, it's like trying to find a needle in a haystack without knowing what the needle looks like, right? So if I know what the needle looks like, I'm ahead of the game. And also, and then the machine can help me. But we still need to have hypotheses, have good minds, think about what the right data design, study design would be, and how we could make these fatal mistakes where we are thinking it's the stork that's bringing the babies, right? Or the yellow finger of that causes the lung cancer, the yellow finger of the heavy smoker, right? Because it's very heavily associated. It doesn't mean it causes it. It has one common cause, which is smoking. So we still need brains.
Okay, so just 5 minutes, and I have seen some people, so no more questions at this point. I know who I'm going to. Thanks.
Hi, you were mentioning that rotenone is marketed as an organic agent. So does that mean it would be found in organic food products?
It has been used by organic farmers, and I know because we were actually approached by a couple that both developed Parkinson's in their 50s who were organic farmers, and this is anecdotal. I have no proof that that was what caused their disease, but it was a couple and they were organic farmers and they said they had used rotenone. Probably heavily used rotenone, if you ask me. But yes, it was, you could find it in garden stores, and people put it on tomatoes. It's also used as to kill invasive fish species. So that is the main use actually more broadly. So it's put in waterways to kill invasive species.
So are there any research studies showing that people eating an organic diet have lower rates of Parkinson's or slower progression?
No, we have no studies like that.
Okay, thanks.
Unfortunately.
So now we can drink coffee and exercise and so forth to keep the thing from progressing. What about DBS? Have you done any studies on the effect of DBS in progression?
Deep brain stimulation.
Yeah. Well, I think that's not really data that's out there yet. But I know that people who have deep brain stimulation are being followed. So hopefully at the Movement Disorder Society meeting the last day where they are showing the latest, there will be some new data, but it's not out yet. Not that I know. But I wouldn't really think that it's protective. It's because it's more of a symptomatic intervention. It curbs the symptoms. I don't think it really helps you not to progress. Yeah. Yes, you can do that, but I would be very careful in interpreting it. Yeah, it's really a risk, not, it's not a one-to-one relationship.